Hello guys and welcome back to Excelsure Academy. So yesterday we did certain MCQs based on the previous lectures, right? So now we'll move forward and today we'll discuss about the solute transport. Okay, so under the category of membrane solute transport, many things are there, and we need to discuss about the osmosis, passive diffusion, active diffusion. All those we are going to study under this category. Okay, so now across a membrane, so we are going to transport molecules across a membrane, yani ki from exoplasm to the cytoplasm or vice versa. So the transport process are critically important for all the living cells because all living cells necessarily exchange the substance with their surrounding like nutrition, waste, toxicant or any inorganic electrolytes, right? Now, the membranes are selectively permeable. So what does it mean? It means it will not allow all the molecules to pass through, right? So depending upon its composition, whether it is a small molecule or it is a lipid soluble or it is hydrophobic in nature, depending on its composition, it will allow. Now, for example, this is the category, this is the table list which is listing the molecules which it will allow and which it cannot pass through the membrane. Okay, so water, yes, carbon dioxide, oxygen, and other polar molecules such as ammonia and lipids such as cholesterol. So these are the molecules which can pass through the membrane easily. Okay, they don't need any what to say, the carrier proteins or any channel. We'll talk about water later. So like water does have channel also. Okay, even though it can go directly, but it sometimes needs channel also. So we'll discuss about this. This is the exception. Now coming to the chemicals that cannot pass through the membrane. So all ions, including the hydrogen ions. Now why? Because of their charge. Okay, because of the charge, these ions cannot pass through the membrane. Now, if it is mid to large polar molecules, including glucose, so even that cannot pass through the membrane. Okay. Now, for example, molecules, polar molecules like glucose, okay? So glucose, since it is bigger in size, cannot pass through the membrane, and the membrane has got here the hydrophobic part, right? And we know glucose is hydrophilic molecule. So it cannot pass readily. So it needs again channel. Then amino acids, macromolecules like proteins and polysaccharides. So whatever which is bigger in size cannot pass through the membrane. Simple. Now, what are the different types of membrane transport which we will be talking about? So passive transport includes your simple diffusion, osmosis and facilitated diffusion. में आएगा आपका simple protein channels, ungated gated channels. Okay. Now coming to the active transport mechanism. So what is the main difference between the passive and the active? The usage of ATP. ठीक है. So passive transport does not need energy. It is depending on the concentration gradient it is happening. For example, अगर यहाँ पे आपका concentration high है, यहाँ पे low है, so diffusion is going to happen in this direction. Solute from high solute concentration to low solute concentration. Okay. Now, in the case of the active transport mechanism, even if the concentration gradient is there, like let us say that it is high over here and low over here, but the requirement says that the molecules or the ion should pass from low to high. So since it is against the concentration gradient, ATP is required. Okay. So active transport via the protein pumps, bulk flow mechanisms like endocytosis. Under endocytosis, we have got the phagocytosis, pinocytosis, and exocytosis. So all this we are going to cover. And here also many different types of channels are there. So there are, like, for example, we are going to talk about the Na plus K plus ATPase. We are going to talk about the F type, B type, and P type pumps we are going to talk about. Then there is something called ABC transporter. So all these categories we have to cover, and at the end, so once we reach this step where we are going to discuss about the endocytosis and exocytosis, then you can conclude that the membrane transport is getting over. ठीक है, तब तक तो हमें बहुत सारे transport पढ़ते ही जाना है. Okay, so first let's start with the osmosis. So osmosis क्या है? Simple. It is the diffusion of water molecules from more dilute solution to a more concentrated solution. ठीक है? यानी कि क्या? High solute to low solute, और अगर water के terms में कहना है तो जहाँ पे high water concentration होगा to a लो कॉन्सेंट्रेशन ऑफ वाटर, ठीक है. सो इट इज अ स्पॉन्टेनियस प्रोसेस. जब भी ऐसा ग्रेडियंट बनेगा आपका हाई टू लो वो मूव करेगा. नाउ द प्रेशर रिक्वायर्ड to maintain the equilibrium where the net movement of solvent is zero, it is known as osmotic pressure. Please guys, please understand what this means. ठीक है. So अगर suppose आपका diffusion हो रहा है, ये आपका membrane है, and the net movement of the solvent is stop, यानी कि ये equilibrium में आ गया है. So the pressure which is required to maintain this equilibrium is known as the osmotic pressure. Is it clear to everyone? Yes.
Yes, ma'am.
Okay.
Yes, ma'am.
Simple concepts, right? Same thing here again. So osmosis is there. You have got a semi-permeable membrane, and then you can see it over here. So what do we see? The concentration of the molecule is over here high as compared to here, right? So here the water concentration is high. So they will keep on moving till it becomes the अंडर इक्विलिब्रियम. सो यानी कि जितना यहाँ मॉलिक्यूल है उस रेस्पेक्ट में वहां पे उतना पानी होना चाहिए. सो इवन इफ यू सी दैट ओवर हियर द level of water is high but the amount of solvent, okay, and the solute respectively has been increased. ठीक है. तो अगर यानी कि यहाँ पे 1 लीटर है और यहाँ पे आधा लीटर पानी है तो रेस्पेक्टिवली उतना ही सोल्यूट प्रेजेंट है. यानी कि क्या, the molarity over here and the molarity over here is going to be the same. Okay. Now transport and transfer across the cell membrane. So this also we know it's very basic. Okay, I'm just reading this. Isotonic means having the same osmolarity. Hyper means having the higher. Hypo means less. So if you put a cell into a hypotonic solution, it is going to burst. ठीक है. Now if you put into a hypertonic, यानी कि salt concentration आपका बाहर ज्यादा है, so it is going to shrink or shriveled. Okay. Simple. Isotonic means nothing is going to happen. Now let's talk about the turgor pressure also. So the turgor pressure, it is the hydrostatic pressure in the cell wall. Now this turgor pressure comes with respect to your plant cell only. ठीक है, because animal cell does not have cell wall. So plant cell में आपका cell wall present है and then cell membrane is there. So we need this turgor pressure in order to maintain the shape of the plant cell. Okay. Now the cell wall enables cells to take extra amount of water without bursting. Okay, so that is the advantage of having a cell wall. Now without bursting the cells, take in water and push against the cell wall, which pushes back many cells use which pushes back. Now many cells use turgor pressure as part of maintaining the structure, and if they lose turgor pressure, the plant starts wilting. So you must have seen the plant, एक खिलखिलाता हुआ लीफ होगा और एक मुरझाया हुआ. So that is all because of the turgor pressure if you have seen. ठीक है. Okay. So any doubts so far? No, I guess, right? Because these are very simple. Yes, any doubts are there? Akshaya, Tanvi, Shruti?
No, ma'am.
Okay. Now let's talk about the passive transport. Okay. So passive transport, already as mentioned, that it is the transport of molecules down the concentration gradient without using the energy, that is ATP. So simple diffusion, again same thing. To move from high to low without a protein channel. ठीक है. तो यहाँ पे क्या होगा, आपको जो भी molecules है from high concentration to low concentration it is moving. Okay. Now it is a transport of ions of molecules from high to low concentration without energy expenditure across a membrane. So no energy, no protein is required, and molecules like N2, nitrogen gas, oxygen, CO2 can easily move. Okay. Now from here onwards, guys, start giving more concentration. Now, because the transportation of such uncharged molecules across the membrane is an entropic process. Okay, so we have already mentioned that it was a spontaneous process. So spontaneous कब होगा? When it is going to be an entropic process, right? Delta S आपका ज्यादा रहेगा. So in which the movement of molecules happens from high concentration to low till equilibrium is reached. Now let us say that C is the concentration of salt at higher concentration and C low is a concentration of solute at low concentration. ठीक है, on one side of the membrane. तो अगर ये मेम्ब्रेन है, एक जगह पे आपका C हाई होगा और एक जगह पे आपका C लो होगा. Okay, now how do we give with respect to the free energy? So delta G is equal to minus RT ln natural log, then concentration of C high to the concentration of C low. ठीक है. Now R क्या है आपका गैस कांस्टेंट, T हो गया टेम्परेचर इन केल्विन. Now if you want to make it to the logarithm to the base 10, okay, from natural log to log 10, then आपका ये वैल्यू ऐड हो जाएगा. ठीक है. So now delta G is equal to minus 2.303 RT log C high to C low. So the difference in the concentration of high and low is termed as a concentration gradient, and delta G here it is known as the chemical potential difference. अगर मैं बोल रही हूँ कि membrane के पास यहाँ पे high and यहाँ low है, so the energy which the membrane possesses is known as the chemical potential difference. Is this slide is clear to everyone? Anywhere any doubt is there which you want me to clarify? Tanvi, is it clear?
Yes, ma'am.
Okay, good. So what you have to remember here is this formula. Okay. Now, when I say, so I'm just going to repeat one more time. So यहाँ पे अगर आपका membrane है, you have got the concentration here high and here concentration low. So because of this concentration difference, okay, a chemical potential difference is created. This is nothing but energy. उसमें वो energy है. So जब आप इस चीज को equilibrate करते हो, so what is going to happen? The molecules are going to move from high to low. Correct? So this chemical energy can be converted into mechanical energy or electrical energy. वो हम आगे जाके देखेंगे. ठीक है? Now, so this can be written in this form. कि कितना chemical potential difference is present at the membrane can be calculated using this. Okay. Now, अभी कैसे होता है? For example, like if you know those neuroscientists are there, right? So they are working on the neurons, right? And neurons आपको impulse देता है, electric impulse देता है. Those are nothing but the firing of the neurons, यानी कि वो आपको signal दे रहा है. And these signals are understood by the brain in order to give the response. ठीक है? So ये signal, ये impulse, ये क्या चीज है? This is nothing but the concentration difference in the ions in respect to Na plus हो गया या K plus हो गया या Cl minus हो गया या calcium 2 plus हो गया. So इनके difference के वजह से वो impulse generate होता है. ठीक है? So that is the potential difference created over here when a neurons are firing a impulse. Just giving you a gist like how neurons are working. So keep in mind this thing because from here onwards everything is being it is important for you to understand like how membrane is getting how membrane is helping in the transport of the molecules. Okay. Now, so okay. So now passive transport of the charged molecule. So there are two types are there. So that can depend upon the two factors. Okay, movement of the charge ions. So electric voltage gradient or chemical concentration gradient. Okay. So if suppose both are forming, okay, chemical concentration and the voltage difference is coming. So it is known as the electrochemical gradient. Okay. So that determines the energetically favorable direction of the molecule transport. अब इस पे depend होगा then only the molecules which is at low and high, depending on this, the direction will be determined. ठीक है? Okay. Now, if suppose अब तक तो हमने uncharged molecule की बात की. Now, when we talked about charged molecule, for example, Mg2 plus हो गया, right? या K plus हो गया. और what else we have NH4 plus हो गया और phosphate ion हो गए, right? So when you have got such charged species with you, then how it is going to get affected? So here Z is the charge on the transported species. F is the Faraday's constant, which is given as 96485. Okay, and then delta V. So delta V is the electric potential difference across the membrane. So the equation what we have just learned now get improved when you have a charge species. ठीक है? So plus or minus. So plus कैसे जब if you have got cations, minus if you have got the anions. Simple. Okay. So this is the another important point. This is your another important point which you have to remember. Okay. Now look at the diagram. So how the electrochemical gradient is formed. This is what we are going to see now. Okay. So in one direction, in one side of the plasma membrane, you see that the concentration of Na plus is high. Okay. And here it is low. And that is what the arrow मांगी जो बना है आपका. So high over here and it is low over here. Right. Now with the passage of time, the Na plus concentration here and here becomes same. तो ये आपका इक्विलिब्रियम में आ गया है. ठीक है. Now ये जो पोर्शन हुआ यहां से लेके यहां तक का टाइम, so that time if the membrane was acting as a electrochemical gradient. Is this much clear, guys? Once it reaches equilibrium, then it does not have the gradient. ठीक है? तब आपका gradient नहीं रहता है, it becomes normal. Is it clear? Are you able to follow? Akshay, are you able to follow?
Yes, ma'am.
Okay, good. Now, so the passive transport always depends on the following factors. So you have got the substrate concentration across the membrane, right? So first is the substrate concentration. It can be your ions or whatever. Now, hydrophobicity of the molecule to be transported. If the molecule is highly hydrophobic, it is easy. If it is hydrophilic, then it is difficult. The size and the charge of the molecule. If there is no charge, easy. Small size, easy. Bigger size and charge, it is difficult to cross. Okay. Then the electropotential difference across the membrane and the diffusion area and the distance. इसका क्या मतलब हुआ? Simple. If you have got smaller area, ठीक है? इसमें इतने area में diffuse होना is faster as compared to the diffusion rate over here. Obviously, right? जितना ज्यादा एरिया होगा उतना ज्यादा आपका टाइम लगेगा डिफ्यूज होने में. Are you getting the answer? Getting the point over here? So if you have small area, the diffusion rate is going to be high. Larger the area, diffusion rate is going to be the less, क्योंकि उसको उतना टाइम लगेगा पूरे एरिया में स्प्रेड होने के लिए. ठीक है? So Shruti, is it clear to you all the 5 points?
Yes, ma'am. Yeah.
Okay. Okay. Please feel free to ask, okay, even if you want me to repeat. Okay. Now here, this is very important. This has been asked in the exam. So hydrophobicity of a substance is measured by its partition coefficient. Now what is partition coefficient? It is a partition between the oil and water. अगर कभी ध्यान से देखो, so suppose this is your water, okay, and here on the top you have got the oil. Let us say that oil is there. ठीक है? Now the space between यहाँ पे between the water and the oil droplet, यहाँ के जो इनका जो contact है, that is the partition. Okay. See, since it's oil and this is water, it is not going to mix. They are immiscible, right? So they have got the partition where this is a layer of your oil and this is a layer of the water. So this thing is known as the partition. So here the K is the equilibrium for its partition between the oil and water. So partition coefficient K. Okay. The substance with high partition coefficient is more soluble in lipid. यानी कि क्या? पार्टीशन कोएफिशिएंट जितना ज्यादा होगा, ये द डिस्टेंस जितना ज्यादा होगा, इट मींस इट इज हाईली लिपिड और हाईली हाइड्रोफोबिक इन नेचर. ठीक है? सो इट विल बी मोर सॉल्युबल इन लिपिड. आर यू गेटिंग द पॉइंट? जितना ज्यादा आपका हाइड्रोफोबिक होगा उतना ज्यादा लिपिड सॉल्युबल होगा. इज दिस मच क्लियर? ओके. नाउ फॉर एग्जांपल, डाईईथाइल यूरिया having an ethylene group attached to each nitrogen atom of urea has पार्टीशन कोएफिशिएंट ऑफ 0.01. डाई इथाइल यूरिया का फॉर्मूला क्या है? एनीवन? अच्छा, यूरिया का मालूम है किसी को? द स्ट्रक्चर ऑफ यूरिया, एनीवन? CO, यहाँ पे NH2 आ जाएगा और यहाँ पे NH2 आ जाएगा, ठीक है। अगर मुझे डाई इथाइल यूरिया करना है तो यहाँ पे एक H हटा के इथाइल ग्रुप आ जाएगा, ठीक है। यहाँ पे एक H हटा के इथाइल ग्रुप आ जाएगा, ओके। ना लुक एट ओवर हियर. So the partition coefficient K of diethyl, sorry, हाँ, diethyl urea is 0.01, okay. Whereas only urea has, अभी यूरिया क्या था आपका? CONH2 and here NH2, ठीक है. So what is the K over here? 0.0002. Now tell me which has got the higher partition coefficient. Is it urea or diethyl urea? Tanvi? किसका ज्यादा है?
Diethyl urea.
Correct, absolutely correct. So it has got higher partition coefficient, okay. Now since it has got higher partition coefficient as compared to urea, it is more hydrophobic in nature, okay. Now अभी क्वेश्चन अगर आपको ये पूछे कि डाई इथाइल यूरिया और यूरिया में कौन सबसे इजीली डिफ्यूज होगा थ्रू प्लाज्मा मेम्ब्रेन, सो व्हाट विल बी योर आंसर? यस, एनीवन? अक्षया?
Diethyl urea.
Yes, absolutely correct. So it is going to be your diethyl urea because it has got the higher partition coefficient. Partition coefficient ज्यादा होना मतलब it is more hydrophobic in nature. Hydrophobic in nature, अगर उसका hydrophobicity ज्यादा है then it is easy for it to cross through the plasma membrane. Okay, so I hope this concept is clear to you everyone, and when in future this question is asked, please get the correct answer. Okay, time नहीं लगना चाहिए. Okay, now let's talk about the facilitated diffusion. So till now we were talking about the simple diffusion. अभी हम बात करेंगे facilitated. Facilitated diffusion मतलब क्या होता है? Where you need the help of proteins, ठीक है, in order to transport molecules from one membrane to the another membrane. Okay, so facilitated diffusion of uncharged molecule and polar molecule. So it is a spontaneous process. Again, this depends on the concentration gradient only, okay. And these protein or the membrane जो भी है आपके, they are nothing but again the integral proteins which contains the multiple membrane spanning alpha helices, okay. They facilitate net movement of solutes only in thermodynamically favor situation. यानी कि क्या, डेल्टा जी आपका नेगेटिव होना चाहिए, और डेल्टा जी नेगेटिव होना मींस दिस many times I have repeated, delta G should be negative. Now negative means it is spontaneous, ठीक है. स्पॉन्टेनियस है इसका मतलब वहां पे आपका कॉन्सेंट्रेशन ग्रेडियंट होगा. स्पॉन्टेनियस है तो उसको एटीपी की जरूरत नहीं है, ठीक है. These are the points. Now they show a measurable affinity and specificity for the transported solute. So what does it mean? So आपका यहाँ पे plasma membrane है, here you have got the protein, okay. So these proteins have got affinity for the molecules which they are going to transport. ऐसा नहीं है कि कोई भी आके कहीं से भी चला जाएगा, ठीक है. इट्स जस्ट लाइक यू हैव टू शो द आइडेंटिटी कार्ड टू द रिसेप्टर और द प्रोटीन चैनल, देन ओनली इट विल ओपन गेट्स फॉर यू. सो उसी तरह से फॉर द चार्ज मॉलिक्यूल्स वी हैव गॉट लाइक Cl माइनस के लिए अलग आयन ट्रांसपोर्टर है, Na प्लस के लिए हमारे पास अलग है, K प्लस के लिए अलग है. So it's not like that Na plus K plus single charge hai, so either of them can pass through. No, it is not like that. Okay, Na plus ke liye different hai, K plus ke liye separate hai. Similarly you have got for calcium, similarly you have got for Mg2 plus. So jitne bhi requirement hai, every ion or every molecule in the system which goes inside of the cell or outside of the cell has got its own channel. Okay. Now these protein also show the behavior of saturation regarding their binding with solute molecules that experimentally distinguish the facilitated diffusion with simple diffusion. तो ये sentence का मतलब क्या हो रहा है? So you must have studied in your enzyme kinetics that if you have got the enzyme, right? हर एक एंजाइम का अपना सबस्ट्रेट होता है, right? Okay. So what will happen if the substrate concentration increases? Suppose let's say that यहाँ पे आपका substrate concentration है and यहाँ पे आपका enzyme activity है. So what will happen? Shruti, any thoughts? Akshaya?
It will increase gradually and then it will saturate.
Absolutely correct. Very good. So what happens is that as the substrate concentration is increased, the enzyme activity is also increasing. And after a time, it becomes saturated. Now, why? Because the substrate concentration is increasing, right? The enzyme is fixed. So जब तक आपके free enzyme से they got bound to the substrate, so there was increase in the activity. Now, after some time, what will happen? Even if you increase the concentration of the substrate, but the amount of enzyme is going to remain same. Hence, it reaches saturation. In the same way, plasma membrane है, यहाँ पे आपका receptor है, or protein channels, whatever it is. So, if you have got 3 ions to be passed, they will bind and they will pass. But if you increase to 30, still it has reached its saturation, right? क्योंकि वहां पे तो सिर्फ 3 ही है to transport. They have to wait so that this channel can transfer from one place to another. When it gets free, then only the other ions can come and bind. So similarly, like enzyme substrate, it reaches a saturation. In the same way, the proteins also reaches their saturation. Okay, is it clear? This is the
Yes,
same
ma'am.
thing. Yeah. So concentration gradient is there. These are the channels. Okay. These are the channels by which they can bind, they can come inside. So what is happening? Eventually, these channels are helping in making the equilibrium in the concentration. Now, this is very important. This graph is really very important. So what this graph is saying? So here you have got the concentration gradient and the transport rate. कैसा होता है? यहाँ पे solute था पहले, substrate था और यहाँ पे आपका enzyme activity था. Okay. Similarly, here we are having the concentration gradient. Here we are having the transport rate. That is percentage of maximum velocity achieved. So if it is a simple diffusion, it doesn't matter, right? क्योंकि जितना ज्यादा आपका होता रहेगा diffusion आपका वो बढ़ता रहेगा. अगर आप यहाँ पे concentration बढ़ाते रहोगे across the membrane, gradient बना रहेगा, आपका diffusion चलता रहेगा. ठीक है? Now, what happens in the facilitated diffusion? After a point, see concentration gradient आप कितना भी बढ़ा लो, but after a point it is going to reach its saturation because you can't increase a number of proteins which is already present. ठीक है? If it was 100, then only 100 proteins can get involved in the transporter. Okay, is this much is clear? Now, are you able to understand the graph between the simple diffusion and the facilitated diffusion? Yes, are we good? Can we move forward?
Yes, ma'am.
Yes, good. Now, so many proteins facilitates the diffusion by forming a protein line pathway across the membrane. allowing the movement of hydrophilic molecules without coming in contact with the hydrophobic core of the plasma membrane. So what it is saying is that in the plasma membrane, when these integral proteins are there, right? अभी हमने जो mention किया, so they are actually alpha helices. So they make in such a way that they form a hollow-like structure. जिसकी वजह से क्या होगा? They can just pass through the membrane without touching the हाइड्रोफोबिक रीजन ऑफ द प्लाज्मा मेम्ब्रेन जो यहाँ पे प्रेजेंट रहते हैं. ठीक है. सो वेरी इजीली ग्लूकोज हो गया और एनी अदर polar molecules are there, so they can then easily pass through the plasma membrane. Okay, yeah. So they are known as the transporters. जो मैं भी channels कह रही थी, they are known as the transporters. Okay, for example, they're also known as carriers. They move wide variety of ions and molecules across the membrane. So here you have got the two conformations. So guys, slowly we are just reaching that level जहाँ पे थोड़ा सा complexity बढ़ेगा. ठीक है. So this is a simple example of the glucose transporter. So as you can see here, the two conformations are there. Okay. So what are the two conformations? Close and open. So this is your open conformation. यहाँ पे क्या हो गया? आपका ग्लूकोज है, इट गोज टू द बाइंडिंग साइट ऑफ द ट्रांसपोर्टर. सो दिस इज ग्लूकोज ट्रांसपोर्टर. तो वो वहां जाके बाइंड करेगा. व्हेन द बाइंडिंग इंटरैक्शंस आर करेक्ट, then only there will be a conformational change. So conformational change, what it is doing, it is getting closed over here and open over here. And once it gets open, the interactions are broken between the glucose and this transporter. Glucose is free now to go inside of the cell. Clear? Then again it is closed and open for the next round of transport. Is this much is this figure is clear to everyone?
Yes, ma'am.
Okay. This is how when you eat something, it gets digested and from your stomach it goes to the intestine, right? Now in the intestine, you have got these transporters present. Intestinal cell may, like for example, let us say that we have got over here like this. Okay. यहाँ पे ये intestinal cells हैं. Here you have got these glucose transporters present. So जैसे ही आपके blood में glucose level ज्यादा हो जाता है, okay, insulin is secreted. It gives signal to release to make or the expression of the glucose transporters. So they get expressed on these cells. तब आपका glucose enter होता है. ठीक है. Similarly, you have got different types of transporters for muscles, for brain. Okay, we'll come to that slowly, don't worry. Okay, so these are the types of the transporters: uniporters, symporters, and antiporters. 1 second. Okay, so uniporters, as the name says, it is going to transport single type of molecule down its concentration gradient. As example is glucose. ठीक है. Symporters में क्या रहता है? This happens in the case of like when remember like जब भी कोई faint होके गिर जाता है, so we give them saline or we give them ORS. And if you see the ingredients of ORS, इसमें हमेशा क्या रहेगा? नमक और ग्लूकोज रहेगा. Right? So why it is important is because see, when you need instant energy जो आपको glucose देता है, right? So it is not easy for the glucose to get absorbed fast. So कैसे होता है? Along with the Na plus, the glucose will be taken inside. For example, SGLT1 is the transporter name. तो यहाँ पे Na plus भी साथ में जाना चाहिए and आपको glucose भी साथ में जाना चाहिए. So this is the reason why when someone faints or having low glucose level in their body, we always give Na plus, NaCl also. Okay, so this is the reason why the glucose and the Na plus NaCl is given together, because it increases the absorption of glucose in the body. Clear? ये concept clear हुआ?
Yes, ma'am.
Okay. Then we have got the antiporter. So antiporter same as it says. So if you have got the transporter like this, so Na plus यहाँ जाएगा तो Ca2 plus बाहर जाएगा. So if suppose something is coming inside, the another molecule is going to go outside. So each category we are going to discuss in detail with different examples. Okay. Yeah. Okay. So now let's take this example of GLUT. Okay, so this is a glucose transporters, and we have got 12 types of glucose transporters, and it has got, each glucose transporter has got 12 hydrophobic membranes spanning alpha helices. So what does it mean? In a plasma membrane, so we have got 12 times like this. Okay, so ऐसे 12 times रहेंगे. So amino acids like serine, threonine and glutamine, asparagine, they forms hydrogen bonds with the OH group of the glucose. So remember I was telling you that in glucose it goes and makes interactions, that is a binding pocket, right? So in the binding pocket it is going to make interactions with amino acids like serine, threonine, glutamine, asparagine. ठीक है, तो ये आपको याद रखना है in the case of the glucose transporters. So this is one important point that you have to remember. Okay, now all GLUT proteins transport glucose, but their differential expression among the various body cells enables them to regulate glucose metabolism independently at different rates. So what does it mean? See, the requirement of glucose in muscle cell is different as compared to the brain cells, as compared to your heart cells, as compared to your other body parts, right? So this requirement varies for each body part, and that's why there is a differential expression. Okay, for example, GLUT1 and 3 are found in RBC, where you have got the high rate of glucose uptake. Then GLUT2 is found in liver, high influx efficiency in response to insulin, which is converted to glycogen. Okay, clear? So when the body gets a signal, okay, there is so much of glucose present in the blood, right? And it is not required now. अभी हमें इतनी एनर्जी की जरूरत नहीं है, but you might required in future. तो हम क्या करेंगे? एक्सेस ग्लूकोज को ग्लाइकोजन में कन्वर्ट कर देंगे. ठीक है, and that's how the story of becoming fat starts. Okay, we'll come to that later. Now, for example, another example, GLUT4, it is expressed in your fat and muscle cells for glucose uptake upon insulin response. Sorry. So here you need to understand is that depending on the requirement, हर एक body part, हर एक cell का, the requirement of glucose is different, and that's why there is going to be difference in the differential expression. Okay, so brain requires more glucose as compared to the liver or as compared to the muscle. Okay, for example, if you're running, so your muscle cells are more in action, so that time the uptake of glucose will be more as compared to the rest. Okay, so as your body perform any kind of function, it is going to have the difference in the expression. Now this is the same diagram, just represented in a different way. So you have got the GLUT transport, glucose binding site is there, glucose comes and binds over here. See, glucose comes and binds. ठीक है, अंदर गया, बंद हुआ, it goes outside, the glucose comes inside, and then again back to this side. So this is just that the conformation, different conformation of your GLUT. There are two conformation only, open and close. ठीक है, close में क्या होगा, आपका glucose bind करेगा, open में it is open, that is either this side it is releasing or this side it is releasing. Clear? Am I going fast? I hope you guys are able to follow. This is very simple. Okay, इसके बाद complicated वाले आएंगे. So I hope Shruti, Akshaya, Tanvi, you are able to follow.
Yes, ma'am.
Okay, good. Now, since I said that these are 12 times alpha helices, okay, so this is how it looks like. Can you see? So here the N terminal starts, rotate, 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 and comes back. So what does it mean? So in the case of glucose transporter, N terminal and C terminal are on the same side. कभी कभी क्या होगा, एक आपका मेम्ब्रेन के उस साइड होगा और एक दूसरा आपका इनसाइड में होगा. ठीक है. Now if you see the overview on the top view of your GLUT transporter, you can see that they are forming a pore like structures. ठीक है. तो अब क्या होगा, ये जो सेंटर जो कैविटी है यहाँ पे आपका ग्लूकोज आके बाइंड करता है, and these are the alpha helices which is making interactions. For example, just now we have mentioned serine, threonine, asparagine and और कौन सा था, glutamine. ठीक है, so these are the amino acids which is making the interactions with the glucose, and once it sorry, once it confirms that it is glucose only, then only it transfer to the another side. क्यों, अभी क्यों कंफ्यूजन आ रहा है, क्योंकि ग्लूकोज जैसा तो आपका मैनोज भी दिखता है, गैलेक्टोज भी दिखता है, दे आर दी एपीमर्स ऑफ ग्लूकोज, just there is a difference in the H and OH arrangement, right? But the condition for GLUT transporter is that it has to be glucose only. अगर कोई दूसरा एपीमर आ रहा है, it shouldn't, नहीं तो ब्रेन को आप ग्लूकोज दे रहे हो, गैलेक्टोज दे रहे हो, मैनोज दे रहे हो, क्या दे रहे हो पता नहीं. So that can create chaos, okay, in your system. So it is very important for the GLUT transporters to make sure that it is transferring glucose only. Okay, now look at over here, this figure. So here you can see that they are arranged like this. Okay, I should have some other marker, one second. Okay, so this is like open. Now this is your close because glucose is there. यहाँ पे देखो, यहाँ पे आपको ग्लूकोज बाइंड होगा, देन लिटिल कन्फर्मेशन चेंज इज देयर. व्हाई, बिकॉज जो ग्लूकोज यहाँ पे जो है इट हैज टू कम आउटसाइड, राइट, एंड देन यहाँ पे ये ऐसे ओपन हो जाएगा. सो एबल टू फॉलो outward facing, inward facing, correct? So this is how glucose is transported with the help of the GLUT transporter. Now this is the scenario where we were talking about the symporter, correct, or co-transporter, where along with the glucose Na plus is also required. Okay, SGLT1 the example. So कैसे होगा, जैसे ग्लूकोज को एब्जॉर्ब करने के लिए, see what happens over here, glucose is there, Na plus is there, both have been taken, right? Then it goes over here, these are the glucose transporter, तो यहाँ से सिर्फ ग्लूकोज ही जाएगा. But what about the Na plus? So Na plus का level inside the cell and outside the cell will be maintained by Na plus K plus pump. This is different. Okay, this is different. So if you see, when in a cell any function is going on, it is not that only one particular transporter is working. Along with that, many antiporter, co-transporter, everything is working. Okay, so once we understand their function, we will be able to understand how it is functioning in the entire cell. So this is one example. I hope it's clear. Okay, so now what is left is about this transporter and this transporter, which we will be covering in the upcoming lectures. So guys, for today, I hope it is clear, right? GLUT transporter कैसे काम करता है? Now, let's talk about the kinetics over here. So the kinetics of glucose transporter with the help of the GLUT1 uniporter is similar to your enzyme catalyzed chemical reaction. So you have got the S out GLUT1, then S out GLUT2. ये क्या हो गया आपका? ट्रांसपोर्टर हो गया, ठीक है, एंजाइम सबस्ट्रेट बन गया है. Then S in GLUT1. तो यहाँ पे एंजाइम कौन है आपका? GLUT1. S out क्या है आपका? ग्लूकोज विच इज प्रेजेंट आउटसाइड, राइट? Now so glucose out with the receptor, with the transporter, it makes the interactions and then it is taken inside. Now if you want to represent like how much velocity it has taken, like what is the catalyzed reaction of the GLUT1, it is given as V is equal to V max S by Km plus S. Normal आपका Michaelis-Menten वाला equation, ठीक है? And people have found that the activity of GLUT2, 4 and 3. So from this figure, you can see that as the concentration of glucose increases, GLUT3 को maximum activity है, ठीक है, as compared to the other. And we know that GLUT3 is used in the brain. So इसका मतलब क्या? The requirement of glucose by the brain cells is high as compared to the other part of your body. Clear? And these are the other GLUT where you can find the primary site of expression, and it also talks about the affinity to glucose. जितना ज़्यादा high रहेगा उतना ज़्यादा plus दिए रखा है, ठीक है? Okay, this is okay if you can't remember, but try to understand this okay, this graph which is important. So इसमें हमें क्या देखने को मिला है, that GLUT3 has got the maximum velocity, okay, at a given glucose concentration. Okay, that's all for today, guys. Any further doubts?